I did present results from our work both in the lab. We have done mouse models, and we have studied imaging in mouse models, and we have also done a phase 1 study with immunopets. So that was part of the presentation. So we had radio-labeled daratumumab with a molecule called zirconium-89, and we first in the lab injected myeloma cell lines in mice, and then we injected this antibody, and we were able to show that the antibody binds to where the cell lines are located, and we could detect this with PET scanners from mice in the lab. We could see the myeloma. So that led us to continue developing this strategy into humans. So we wrote the full protocol for a phase 1 study, and we then submitted that to the FDA and obtained an IND for this. We showed, based on 10 patients, that it's a safe strategy to give this antibody. There are no surprises in terms of new toxicities beyond what you see with daratumumab as a treatment strategy, which is virtually nothing. If you pre-medicate, you virtually don't see any infusion reactions with this strategy either. We further show that the imaging could be done 5 or 6 days after the antibody had been given. If you do it too early, there will be a lot of background noise, and you would have antibody circulating in the blood, and it could be in different organ systems, in the liver and the spleen, etc. So if you wait for 5-6 days, you clearly see if there is focal uptake or not. We also showed in only one patient, because this was the phase 1 study that's designed to study safety and feasibility, but one patient we had done a conventional FDG PET-CT, which is a standard PET-CT that we normally do in the hospital, and everyone is doing the same PET-CT FDG. So in a patient that had myeloma, who was on the study, we did the FDG PET-CT, and there was no focal uptake. And a week later, when we did the immunopet, we saw that there were multiple sites of active uptake in this same patient. We did not, in the phase 1 study, have image-guided biopsy, but when we look at the CT, we saw that there are bone destructions at the same exact site as there is this uptake of the immunopet. So that's very encouraging for potential uptake of residual disease. So overall, we deem it safe, we deem it feasible, and it's very exciting. So we also interpret this as a signal for us to develop a phase 2 trial. So we are now working, and probably in the coming 2-3 months, we hope to open a phase 2 study that will include 50 to 60 patients, where we will compare it to conventional FDG PET-CT. We will also compare it to bone marrow biopsies and blood work with M spikes and light chains to see if this could be a more sensitive technology than conventional imaging, biopsies, MOD testing, and blood work. So we are very excited about it. And this is the first time, to my knowledge, this has been done in patients with multiple myeloma.